Good morning. We are at the 2023 iwCLL meeting in Boston, and we are about to do an informal interview with Dr. Guillermo, formerly from the Pasteur Institute, formerly from Uruguay, and living in France now, permanently?
Living in Paris, yes.
Paris, permanently.
Because my 4 children and 9 grandchildren, they all live in Paris. I'm very lucky to have them all in the same city.
That's excellent. But you started out in Uruguay.
Well, I was born in Uruguay. I lived in Uruguay until 34 years. I did my MD studies in Uruguay, and I came back to France because Professor Binet contracted me for a position, 1-year position, that needed to be renewed. And I refused the renew because my idea was to come back to my country. And when I went to my embassy to renew my passport, it was picked, take off. So I was obliged to rest in France, and I have no chance because no papers. And that is why I rested in France, and I did my career in France, initially in Pitié-Salpêtrière with Jacques-Louis Binet. We always collaborate with Jacques-Louis Binet, and then at the Pasteur Institute in Paris.
Well, as was very nicely pointed out yesterday at the end, the last session by Tom Kipps, and I always pronounce Hospital Saint-Pierre.
Pitié-Salpêtrière.
Pitié-Salpêtrière is, quote unquote, where it's sometimes said that's where it all began. I think maybe it all began in John Louis' apartment and maybe moved to the hospital.
Well, Jacques-Louis, the idea of the meeting, Kanti Rai and Arthur Savitsky and Jean Cronkite have published the initial staging system from RISE, what is called the Ray staging system. And with Jacques-Louis, we started working and we published an alternative staging system. That is the ABC staging, known as the Binet classification. And Jacques-Louis said, why we don't invite these people, that is Kanti Rai, Arthur Savitsky, Jean Cronkite, with somebody else, that is John Galton, and Danny Katowski, to this meeting. And it was a very nice idea. It was a very spontaneous and informal meeting, which was done in the Pitié-Salpêtrière hospital. It was very nice because a cooperation, there are 2 personalities that are very generous personalities and very cooperative, that is Kanti Rai and Jacques-Louis. This relationship functioned very quickly and very well. And it was at that meeting, and Morgan Hansen, that was a Danish. Why he was invited? Because he didn't follow in CLL. And the reason is because he published his MD thesis with the clinical records from 150 patients. And Kanti Rai could produce his staging system studying these records from Morgan Hansen.
I had forgotten that what you just described, what Dr. Rai did was apply the staging system
Yes.
To that data. As an aside, I have Dr. Galton's signed copy from Dr. Hansen of the single issue of the Scandinavian Journal of Hematology,
Yes.
which is where his thesis was published.
It was a fantastic work.
Oh, it's
I don't know why he didn't follow.
I've pursued that question, perhaps not in as great as depth as maybe a real historian would, and even discussed it with Dr. Rai, and Dr. Hansen just went his way, what he was interested in, and in fact is when I first started doing this work and approached Dr. Hansen, he actually sent me. I have 2 copies of his thesis, and he provided me with a photograph, a PowerPoint, and as far as I know, he's still living. I would love an opportunity to meet him, but I don't know if it'll happen.
But Jerry, you knew at that time CLL did not interest anybody because it was a disease, a frequent disease, but chronic disease, and with no therapeutic issues. The only possibility was chlorambucil, sometimes chlorambucil plus prednisone, cyclophosphamide, but there were not drugs. Nobody were going to the sessions devoted to CLL. Everybody was working on acute leukemias, on ingravescent lymphomas, but CLL did not interest anybody.
You know, another aside, in the opening of the meeting, Jennifer Brown, Dr. Brown, had a timeline that she put up, and she started her timeline with Professor Minot, M-I-N-O-T.
And Farber, yes.
And Farber's second one. But she drew attention to Professor Minot because he essentially discovered the gastric relationship to B12. Yeah. But prior to that discovery, he is the first person, and I forget, Minot and...
Yes, in the CML.
They were the first people, the first 2 investigators to treat CLL. And the only drug they had for treatment in 1927 or 1926 was radiation.
Radiation, I remember.
And the radiation worked well, except the disease came right back.
Yeah.
And that was pretty much the end. You know, that makes me think of another question that I've been trying to figure out the answer. You know, there was a period when both Dr. Benet and perhaps Rai, and even when Galton went to, I think he went to Oxford to meet with Gowan to study the lymphatic circulation. He went there because Gowan had learned to make cannulas that could be inserted into the thoracic duct of a mouse. And I think Rai was thinking about working with the thoracic duct on cattle because it was so much bigger.
I know very well the work from Gowan's because Jacques Louis also, he worked and he was cannulating sheep.
Sheep, okay.
In order to study the lymphatic circulation. And Jacques Louis, sometimes he tried a method to irradiate CLL cells by extracorporeal irradiation, making the circulation, and it didn't work. But he tried, and he worked a lot of time with a Canadian student in St. Louis Hospital, cannulating sheep to study
Extracorporeal.
Lymphoid circulation, like Gowan's. Gowan's has made a very important work.
Where Dr. Rai was at that institute where he started out.
Brookhaven. With Cronkite.
Cronkite, I think, also supported the effort for extracorporeal radiation. Yes. The reason I'm mentioning it is that perhaps the next, not many papers, 1 or 2, was lymphangiograms. Why do a lymphangiogram?
We did with Jacques Louis. We have studied and we have related lymphangiograms to the ABC stations. Was published in a radiological review about 40 years ago. And the final conclusion was that it was not bringing too much new information.
And I wonder if that conclusion, which certainly in retrospect was correct, was there much of a lag between stopping lymphangiograms and the onset of CT scanning?
Yes, because it was prior to CT scan,
It was.
this study, and then when CT scan came, it was not lymphangiography is something like a barbarian method.
Yes, yes. But I wonder if the language, the descriptive language that was used to describe the different patterns of the lymph nodes, not so much the regional lymphadenopathy, but the actual pattern of the dye in the lymph node, whether it was foamy or whether it was streaking or whether it was fatty.
Well, in this study we didn't study this because I have co-signed this paper. We did not study this, the morphological aspect of lymph. The only question was whether lymphangiogram could bring new information concerning abdominal lymph nodes. And the conclusion was that it was not bringing too much new information.
Yeah, certainly in retrospect, knowing what we know now, absolutely correct. Well, when I first started this project, and I went to Paris to visit Dr. Benet in his office at the, I think it's like the National Institute of Medicine in Paris. It was.
The Academy, National Academy of Medicine.
And he shared with me the when he first went to, I think, Columbia University, when he first came to the U.S. and he basically started out studying the electron microscopy of, I think
I don't remember the name of the. I think it was a Romanian guy.
I've forgotten the name too.
I forgot the name, but it was initially, and that is why Jacques Louis was interested by electron microscopy. He tried a method that finally didn't work very well. That is a stereological method, three-dimensional, from the picture of the electron microscopy to try to do three-dimensional.
I always like to remind myself with his electron photomicrographs that he has the first description of the dense lymphocyte of CLL, and also he sometimes embarrassingly commented that he also photomicrographs some smudge cells.
Yes.
But it's there for the whole world to see.
Yeah.
And he certainly shared with me that time the process that led to the formation of the Benet staging system. He was quite emphatic in that meeting that this was not a project that was done in isolation. He didn't do this by himself. He essentially kept referring to the French cooperative CLL group.
Yeah.
And at the end of that visit, he really insisted that I make it a point to record something about the whole CLL French cooperative group. And you may recall that was kind of the driving force of why I came to the Uruguayan embassy in Paris to enlarge upon the French cooperative group. And it was quite a large group of people that
Oh,
work.
the French, in the early 70s, 80s, French cooperative group was the most important group in CLL. We have published, there is a paper in the New England Journal of Medicine, I think 99 or something like, perhaps, well, I don't remember exactly, where we produced the results of 1,500 patients followed during more than 10 years. And I think this is one of the most important contributions of the French cooperative group, because this study demonstrated that it was a comparative study in stage A patients between abstention and chlorambucil. And this study showed that there was no benefit from chlorambucil.
Early treatment. And I think that, and I'm glad you bring that up and mention that, because I think that Daniel Katowski made a similar finding with the MRC data.
Yes.
That
Yes.
I think they're almost carbon copies of one another. So those would be two landmark papers. I was just talking with Frida Stevenson in Sussex.
Sussex. She was in Sussex. No, she was close to
Southampton.
Southampton, but it's not Southampton. It's a city outside Southampton.
And the reason I mention it was that the two papers that showed the relationship of the mutational status of immunoglobulins in CLL, those two papers were published back to back in Blood, even though they were rejected by the New England Journal more than 20 years ago.
I will tell you the whole story. Initially, the first paper concerning mutation and unmutated was a paper we published with Harry Schroeder in Immunology Today. 4 years before the papers from Terry, Frida, and Nick Curasi. And this paper was a very difficult paper to publish, because we were saying the initial idea was that all CLL cells should be unmutated. There was an initial fantastic work from Tom Gibbs indicating this. And we were showing that at least half of the patients with Harry Schroeder were mutated. This paper took 1 year to be accepted, and you know what we have been obliged to do in order to be accepted? We have to send the sequences of the patients. We sent the sequences, and then it was very clear. And then I continued working, and I was aware that Nick and Terry and Frida were doing this, and I have exactly the same results in our group. But since then, they started working on this. I wait their publication, and we published 1 month later a letter in Blood confirming these results. Our group published 1 month later.
I have I think this is your
The pathogenesis, yes,
Yeah.
that is a paper.
and this is where you did the sequencing.
Yes.
And fact is, I remember being impressed by the amount of normal cells that you did. I'm glad that you remind me of this. In fact, you got the Ribon A medal
Yes,
at the meeting in Houston.
Yes.
And one of the reasons was for your early understanding of the B cell receptor. And
Because I started my work, my scientific work, by studying the expression of the Ig receptor in lymphoid cells from CLL. And there was a paper in Blood in 1974 where we demonstrated that there was a very low expression of the Ig receptor by CLL cells. We did this with Thérèse Ternang and Jacques-Louis Binet by using immunoperoxidase. You have the paper, yes. And
It
this
was the
is,
first quantitative measurement of.
well, it was mentioned in one previous study from the group of Seligman, but it was the first quantitative study because we quantified and we demonstrated that there was a tenfold decrease in the expression of CLL B cells. I would say it's probably the first one, yes. And then we studied this question, and there is a recent paper we produced in my lab at the Pasteur where we have shown the reason for this. And the reason is when you compare intracytoplasmic expression of the Ig and membrane expression, intracytoplasmic is okay, is correct. And then the expression, the membrane expression is decreased. And the reason is because there is a glycosylation defect. It was published in Blood 15 years ago.
So the glycosylation defect inhibits it from being inserted into the membrane.
From being
processed more into
transported to the membrane express.
And yet.
And that is why then it accumulates in the cytoplasm.
And yet it's, and correct me if I'm wrong, but the CLL lymphocyte can still secrete small amounts of the pentameric IgM, and maybe that's what was a block to anti-idiotype treatment for CLL?
Yes, because since that is a puzzling question, because as you know, therapies now are directed against the B-cell receptor, what you call the BTK inhibitors, and they are effective. But if you study the response of the B-cell receptor from CLL when you stimulate with anti-Ig antibodies or with whatever the response is poor. Then why an inhibitor of BTK is so effective? That is a question. And it is because it inhibits BTK, or there is an inhibition of the system of activation of the cell, the whole system. But it is very clear that BTK inhibitors, and this puzzles me.
Yeah, I think that so much has been learned about the role of the B-cell receptor in CLL, particularly now at this level of targeted therapy. Mm-hmm. It's only made more complex by what's going on in the lymph node, in the microenvironment. I sometimes think I spent...
We are in our lab. You know, I continue working in the Pasteur Institute of Montevideo because in 2005 I moved from Pasteur Paris to Pasteur Montevideo to found and create the new Pasteur Institute. And there, one of my students that now is an experiment scientist whose name is Pablo Opeso, who is an Argentinian student. He works very much, and we collaborate, studying the role of microenvironment. And we first published with the group from Frida Steven, with Frida also work on the glycosylation, the effect of the, we are always doing
things
same idea, same thing. And we first published simultaneously. The paper both papers were in Blood at the same date, 2003, that AID activation, the AID enzyme was overexpressed in unmutated CLL when compared to mutated ones. And it wasn't unexpected because since AID, its role is to mutate BH and to induce class switch recombination, why it is overexpressed among unmutated patients? This was a question, a puzzling question.
Rather than the other way around, why wasn't it increased in mutated?
Yes, you will expect that it was increased, but it does not mutate the VH, but it does induce cross-switch recombination. Though we don't know why it cannot mutate unmutated cells. And as you know, AID expression is regulated by the CD40 ligand, which occurs in lymph nodes. And there is a poster from our group in this meeting showing that AID expression is important.
Okay. How are we doing for time?
25 minutes.
Okay. We have about 5 minutes.
We have time?
Yeah. I wanted to ask about how you think about the monoclonal B-cell lymphocytosis as a precursor of CLL.
It's a difficult question. I wrote an editorial in the New England many years ago, and I think Rostom did a fantastic work describing this, but the problem is that if you take aged, normal individuals, the frequency of this monoclonal B-cell lymphocytosis is important, and you would expect this to result in CLL disease, and finally, is quite few, the number of patients. But I think it's an important discovery did by Roston and Peter Hillman.
I think that it's now, I think, fairly well accepted that about 10% of the normal population has MBL.
Yes.
And that in the familial setting, unaffected first-degree relatives, it might be as high as 17%.
Yes.
I'm thinking that cohort of individuals might be worth looking closer at how CLL emerges.
Well, and perhaps this could be, they need more time to CLL occur. That it's such an indolent disease, CLL, that we need to wait. Perhaps we are going to have the answer of this in 20 years. And probably this could be something like a normal event occurring among aged patients, just a phenomenon of aging, as monoclonal gammopathies that occur but we don't know exactly what is going to happen.
I think that if anyone had any doubts about longitudinal samples, cryopreserved, a cryobank, it's going to require those kinds of samples will be useful.
Yes.
Will be helpful. On the other hand, I suppose that you could also take a group of unaffected first-degree relatives and do the natural history. Although I hate to think of how much that would cost, that study.
Oh yes, will take time.
Yes. Well, any closing comments? How are you may I ask how you're finding the meeting in 2023?
Well, I think it's a very nice meeting, very well organized. I think Jennifer did an excellent work, very good. But I was quite puzzled yesterday with these studies on genomics, proteomics, because the technology used is impressive. I remember to do the first sequence in my lab in 5, 8, I don't remember exactly. It took for one single sequence 3 months. Now you can do the whole genome on one day. So these studies are very impressive from a technological standpoint. But I am not impressed by the results. And I think it requires what for me lacks to these studies is questions. Which are the scientific questions that you raise before doing these studies? That is so because we have heard the results. There are very impressive results with a fantastic technology. But it's very difficult to understand why there are 87 driver genes in CLL. What does it mean, all these things? And I think the future will require much more focalized studies by questions, taking groups, homogeneous groups of patients to look what happened with this group compared to other homogeneous, and this is probably the future. And today, it was a very nice meeting concerning treatment in CLL. We are progressing. It's very clear that the new drugs like venetoclax and BTK inhibitors contribute improvement in the disease. But I think cure is not there until.
Have a ways to go yet.